Hey guys, I'm Chesca from Maple Central and welcome back to another video. Today, we're going to talk about phages. Before we start, shout out to Daniella and Natalie who helped me with this video. And if you guys are ready, then sit down, relax, and grab a popcorn because we are taking off. So, phages. What exactly are they? According to Wikipedia, a bacteriophage or phage is a virus that infects and replicates within bacteria. They are composed of proteins that encapsulate a DNA or RNA genome. Phages replicate within the bacterium following the injection of their genome into its cytoplasm. In short, they replicate inside the bacteria, thus killing it. This little creature is one of the deadliest organisms on Earth. Fortunately, we are immune to them, which is why we're still alive when they are inside us. There are two cycles of phage reproduction. First is the lysogenic cycle. The genetic material of the phage, called the prophage, can be transmitted to daughter cells at each subsequent cell division, and at later events can release it. This cycle can also occur in eukaryotes, although the method of incorporation is not fully understood. The first step of this cycle is when the bacteriophage attaches and releases its DNA into the prokaryotic cell. The phage DNA then moves through the cell to the host's DNA. The phage DNA integrates itself into the host cell's DNA, creating prophage. The prophage then remains dormant until the host cell divides. After the host cell has duplicated, the phage DNA in the daughter cells activate, and the phage DNA begins to express itself. Some of the cells containing the prophage go on to create new phages which will help move on to infect other cells. So that was the lysogenic cycle. Moving on, another cycle is the lytic cycle. It results in the destruction of the infected cell and its membrane. There are 6 stages. First is attachment. The phage attaches itself to the surface of the host cell in order to inject its DNA into the cell. Next is penetration. The phage injects its DNA into the host cell by penetrating through the cell membrane. Third is transcription. The host cell's DNA is degraded and the cell's metabolism is directed to initiate phage biosynthesis. Biosynthesis is when the phage DNA replicates inside the cell, synthesizing new phage DNA and proteins. Next is maturation. The replicated material assembles into fully formed viral phages, each made up of a head, a tail, and tail fibers. And lastly is lysis. The newly formed phages are released from the infected cell, which is itself destroyed in the process, to seek out new host cells to infect. These cycles may look similar, but the difference between the two of them is that in lysogenic cycle, the spread of the viral DNA occurs through the usual prokaryotic reproduction, whereas a lytic cycle is more immediate, that it results in many copies of the virus being created quickly and the cell is destroyed. Well, I hope you learned something. Please hit the like button and subscribe below and comment on what content we should do next. Until next time!